So just remember to be vocal if you have a question. So I'm just going to review what we, would have done in class, PCR and sequencing, and then we'll take a look at the assignment more specifically. So remember that we were trying to identify unknown bacteria by using a sequence of DNA. And so we had to extract that DNA from the bacteria by boiling them. Then we put them in a high-speed centrifuge to separate all the cellular junk from the DNA. So all the junk formed a nice pellet on the bottom of the tube, and the clear liquid, the supernatant, contained the DNA. Then we had to set up a PCR reaction to hopefully amplify the correct piece of DNA that we wanted to amplify. We confirmed that by running an agarose gel to see if we have a band of DNA corresponding to approximately 800 base pairs. Technically, the size piece is 779 base pairs, but when you actually do the PCR and sequencing, you really don't get exactly 779 base pairs. If you did get a band, then that meant that you would get a sequence of DNA that you could use to identify, right, the unknown. For PCR, remember there are 4 key ingredients. We need the template DNA that is going to be amplified. We need our special thermostable DNA polymerase that came from a thermophile. We need all the nucleotides, ATGC, and we need primers. Remember, primers serve 2 functions. One, they allow for the specificity of the amplification, because the primers are only going to bind to the gene of interest, right? Because they're specially designed to be complementary to a region on the gene of interest, so that's where they base pair and bind. And because there's a region that's double-stranded, that allows DNA polymerase to hop onto that double-stranded region and start replicating. We have to heat up the mixture first to 95 degrees Celsius to denature the DNA, to separate the double-stranded DNA into single strands. Then we cool it down to about 58 degrees, and that allows the primers to scan the DNA, to wander around the DNA until they find the correct complementary base pairing and stick. Then we need to heat the reaction up to about 72 degrees because we are using a DNA polymerase from a thermophile, so it needs heat to function. So we have to warm up the reaction mixture hot enough so the polymerase will function, but not hot enough to denature and separate the primers from the DNA. So the DNA polymerase will replicate, and then we repeat the process over and over again, 30, 40, 50 times. In our experiment, we only do it 30 times, and that takes about 2 hours, right, to do that. And then in the following class, you would have run your agarose gel to verify that you amplified the correct size piece. And remember, the amplified DNA is technically called the amplicon. And so to sequence DNA, we use the Sanger dideoxy technique. And so it uses the same 4 ingredients as PCR plus an extra ingredient. That extra ingredient is dideoxynucleotides. So you have to set up 4 reaction mixtures. Each reaction mixture has only 1 of the dideoxynucleotides. So remember that a dideoxynucleotide does not have the 3' hydroxyl group, so no nucleotides can be attached to a dideoxynucleotide. So as the DNA polymerase is replicating, every once in a while it grabs the complementary dideoxynucleotide and incorporates it into that new piece of DNA, but at that point, no more nucleotides can be added after. And so, since everything is random, every possible size fragment will be generated in that reaction mixture. Each fragment terminates with a dideoxynucleotide. So all the fragments generated in any particular reaction mixture terminate with a dideoxynucleotide. So that's the last nucleotide of that fragment. We run those fragments on a polyacrylamide gel, not agarose, but polyacrylamide, because polyacrylamide is very, very sensitive. Polyacrylamide gels can separate pieces of DNA that differ by only one base pair. So one pair of nucleotides, and polyacrylamide can separate those bands of DNA. So we turn on the power and the DNA migrates. Again, smaller fragments migrate faster than larger fragments. And we read the gel from bottom to top. Remember that each fragment terminates with or ends with that particular dideoxynucleotide. Since we're separating fragments that differ by only one nucleotide, we read the gel from bottom to top, and that gives us the sequence of that DNA. So our first fragment, the smallest one, ends with a C, right? Because it's in the dideoxy-C reaction mixture. Our next fragment ends with a T, but it started with a C, right? Because it terminated with a T, so that fragment is CT. Our next fragment ends with a G, but it starts with C and then T and then G. The next fragment ends with a C, but it starts with a C, then a T, then a G, and then finally C again, and so on and so forth. So all these bands correspond to the fragments that are shown here. And we read the gel, C, T, G, C, T, T, A, G, A. And that's the sequence of our piece of DNA. Ordinarily, polyacrylamide gels can have hundreds or even thousands of bands. And so we use computers today to scan the gel, and then it spits out the sequence. Yeah, question? You gotta talk to me. Question?
I think it was a mistake.
What was that?
I think it might have been a mistake. There's nothing
Mistake.
like that.
Yeah, someone hit the, yeah, that happened before. So, but that's how we sequence the DNA, using the Sanger dideoxy chain termination method. And as I said, it's easily done today. There are labs that specialize in doing this all day long. You drop off your sample on your way home from work, and the next day they email you your sequence. And they do it for a relatively small price. So, last time I was able to show you the ribosomal database. Remember when we entered the sequence, it identified it as Enterobacter aerogenes, which is the old name. Remember, the newer name is actually Klebsiella aerogenes. The ribosomal database does typically give faster results because it's a smaller database. It doesn't have as much information as the NCBI databases do, and not as many people use it. But let's see if the NCBI database will work. So here's a handout. It gives some background information, and there's a link. So you can click on the link, or you can just type BLAST into your browser, and it should go to that page or give you the option to go there. But let's see. if this will work. Okay, so far so good. So remember, I had some sample sequences that you could play with. So also here are the sample sequences to play with. So I copied this one. So we're doing the nucleotide sequence now. Oops, I have to go back and copy it again. And just as with the protein window, the only thing you have to check is show results in a new window. No other parameters need to be changed. How long this takes depends on how big your sequence is and web traffic. Usually takes a couple minutes, sometimes longer. This morning it wouldn't work at all. There was something wrong with the website. It just wasn't working, so seems to be working now. So we'll give it some time. So we're just patiently waiting. There we go. So again, Klebsiella aerogenes. So that's the new name. If we scroll down, we have Enterobacter aerogenes. So remember, there's many sequences that have been input here over the years. It's all up to scientists to input their data. The database is constantly changing. New sequences are constantly being added. Sometimes old sequences get removed because the scientists discovered maybe they made some mistakes, and so they remove them. But there's a lot of information here. DNA sequences, RNA sequences, and protein sequences, right? The amino acid sequences. So let's see. So this is the protein window. So your first sequence is actually an amino acid sequence. And here's the amino acid sequence. And so they do use the one-letter code for amino acids. And so just FYI, I have provided that code in the supplemental information in case you're interested, so you can see the one-letter code. You don't need to use this, but that's what they use for sequencing purposes. It's just the one-letter code. So you can copy and paste this code into the window. What I tried to do this morning was just to enter in some garbage, right? Just a bunch of random letters. Oops, I got some brackets in there, so it's probably not going to like brackets. And see if this will just randomly happen to match something. You know, it probably won't, but sometimes we get a bad match just to illustrate the process. So let's just see if this can match anything. Sometimes it does, sometimes it doesn't. But I don't have any amino acid sequences lying around to test. So we'll see if we get anything from this. What it often says is no significant similarity found. While we're waiting for that, I'll just randomly try something else and see. And this is where showing results in a new window helps. So what do we get? No significant similarity found. So, which often happens when you type in junk. But occasionally it poorly matches something. So I just wanted to see if this will match anything or not. Nope. Okay. So after you blast your amino acid sequence, you will get a match. And so you need to answer these questions. Remember, there's a separate answer sheet where you put all your answers to submit that through the assignment tool. So how many blast hits were found? So how many matches were found? What's the first organism, the genus and species? And this does occur in brackets with the protein blast. What's the enzyme shown? And so it's important, if you ever get, if your first result is something like unnamed protein product, uncultured bacterial clone, we don't want that. So just skip that, move on down the list. And whenever you first see an organism or an enzyme, then that's the one that you use. And then you have to Google the function of the enzyme. Now, when it comes to making a distance tree, I'm going to show you using the nucleotide sequence because I do have results for that. This is not part of the grade. It's just FYI for you to see how it's done. But has anyone taken Biology 151? So if you're a science major, you're supposed to take Biology 151 after 150. Has anyone taken 151? Nobody's taken Biology 151? Well, if you have, or if you did, one of the things that you did in lab was to make distance trees. So you actually used different criteria and you created the tree on your own, and you saw how complicated it could be. So the NCBI database actually makes what's called a distance tree. It's a phylogenetic tree showing evolutionary relatedness. So I do want you just to take a look at this and play with the sample sequences that I provided to you on Blackboard to see how to make a tree. But I'm going to do this with the nucleotide sequence. So we do have the nucleotide sequence. Find the results. So here, now, one of the problems with these databases is that the web page itself does change periodically. And this is actually very different than what it was to start with. Notice that all of your matches have been selected, so you want to unselect them. And so to make your tree, you choose the first organism that matches, And then you want 4 other organisms. Ideally, you want a different genus. Sometimes the match is so good, there is no different genus, but maybe there's a different species. So choose that. Again, sometimes the match is so good, the genus and species are all the same. So maybe you have different strains, right, that you can use. So we have Klebsiella. Look, here's a Salmonella we can check. Enterobacter we can check. See this uncultured stuff? We don't want that. So, and there may not be any other variety here. No, so no other variety. So maybe look for some, different strains, like ARSS6, here VIT, RSSJ. So it depends on how much variety you have, but you want a total of 5. Let's see how many we have here. So that's 5, right? And then see over here where it says GenBank graphics distance tree. That's the distance tree link that you click. And so this will create a distance tree. Don't want to provide feedback. Now this shows a rectangular tree. So there are 2 things to do here. You see the tools icon, you can click on that. Or if you, oops, that's what I wanted to do, but you can right click and get the same thing. Layout, I did click the slanted cladogram, which is what we're seeing here. And this often is easier to interpret, but it does depend. Notice we have something highlighted in yellow. Anytime in a tree, if something is highlighted in yellow, that represents the original sequence that you input. So you put that sequence in. And of course, according to this tree, Enterobacter aerogenes, strain VITR SSJ, is the closest match because it's right here with our sequence that we put in. But remember, the actual match was this one here, the Klebsiella, the first one. So, trees can be difficult to interpret. They do not always show what you expect them to look like. Something else to do, it's not a problem here, but you see expand all, and that just makes sure that everything displays. Because sometimes when they make a tree, they don't display everything, but in this case they did. But this is our tree, and so it's just to give you an idea. In general, the more closely 2 branches are together, the more closely related the organisms are. So since this was our sequence we put in, this would actually be the closest match according to the tree. But remember, in the results window, it was Klebsiella, right? CMTC strain. So why the tree is not showing that, I don't know, but this is one of the things that happens. And one of the problems with trees is that, it depends on what gene you use or what characteristic you use. You could use a protein sequence, and you may get a very different tree, compared to a nucleotide sequence. So that's one of the problems with distance trees is that you don't get the exact same tree if you look at different characteristics. But this is our tree. It's not very dramatic. Remember, a branch point represents a common ancestor that diverged, right, to give rise to the different branches. We don't want that, but this gives you lots of information here. So, but that's how you make a tree. And it's just for FYI, do not have to turn in a tree. I just wanted you to see how to make a tree because in the assignment, I give you some trees to interpret. So here with the protein sequence, the amino acid sequence, use my trees to answer the question. You do the nucleic acid search. Here's the sequence you're going to enter. You can copy and paste it. No typing required. And again, answer these questions. How many blast hits? What's the genus and species? What part of the gene or gene was used? And you actually already know the answer to C without even having to do any blasting or looking at it, because what's the whole purpose of the experiment that we did, right? And then why is this particular gene commonly used for sequencing purposes? I've already actually given you these answers at least twice. So, but there are reasons why we use this particular gene of interest. And so you need to research that or look through the handouts to help figure out. And you have to give me two things, two answers. That's why it's worth 2 points. And again, you can play with making a tree. Use my trees to answer the question. So again, often a slanted tree is a little bit easier to interpret than a rectangle, right? 'Cause when you have things like this here, it's a bit harder to see that. So that can happen. And the fancy word for these trees is cladogram, a cladogram. This whole thing is worth 12 points. Remember, there's a separate answer sheet, which just has these questions with the answers. So type in your answers and then submit that to the assignment tool, and it's due on Monday. So any questions about the assignment? No questions? So I'm trying to see if I can get something to match. But making a tree is just a practice thing. It's not part of the grade. You don't turn it in. Well, that worked fast, and there were no matches. So. Yeah. Anyway, but you'll use the sequence that I gave you. So any questions? Are we still out there?
Yeah.
Well, blast N is the nucleotide site, and blast P is the protein site. We're not using these others, but you can switch, back and forth more easily here, right, to do a nucleotide sequence or a protein sequence. Or just go back to the start. Then pick, right, which one you're going to use. Let's just see.
When did they create this? When did they start this?
Oh, this has been around for, a long while. I can't think back when. I know it's been around for at least 20 years. It's been around a long time. Like I said, it's periodically updated. So the web pages right now today do not look the same as they did 20 years ago, but it has been a while.
Wow, that's cool.
So let me just expecting that to give me something more than that. Oh, no, it's not doing what I want it to do. Let's see if I can find. Yeah, I can't use a picture though, so I just wanted to get something that I could enter in for you. Okay, here's one I'll take.
So each letter means something else?
Each letter is an amino acid. And so they use a one-letter code as the standard code for these algorithms.
Oh, that's cool. That there's like a standard. I guess there has to be, otherwise everyone would get confused.
Well, right. In order for the algorithm to be able to match and align sequences, everyone agreed on using, or at least the United States said, we're using this, one-letter designation. So that's what everyone else has to use. But usually there's an international committee that agrees on these things, right, to show what, everything should represent. So the sequence that I grabbed from that web page, let's see what it is. Huh. Myeloid differentiation primary response protein from Homo sapiens. So again, the genus and species is in brackets, and the protein here is a myeloid differentiation primary response protein. If this were your assignment, you would have to Google that to find out what it does, but you don't have to for this. It's just an example. So again, if we make a tree, the myeloid here, interleukin, accessory protein. And see, we have a very limited number of matches, so we don't have too much to choose from. So a lot of similarity here. So, and it's all the same. Yeah. All right, so we'll just use these things here. And then here, our distance tree. Our tree, it's shown in the rectangular view, and this one actually looks pretty good, right? The rectangular view. Again, just to be sure you're seeing everything, click expand all, but it was there. If you want to change the format or the layout, right, to slanted, it slants it like this. So, but in this case, either one works well. So remember, the thing highlighted in yellow, that was the sequence, right, that I put in the search window. And it matched myeloid differentiation primary response protein, right? So obviously these are isoforms, so they're basically the same, right? They shouldn't be closely related. And then these two are also related to each other. Again, no surprises there. Remember, the common branch point represents a common ancestor. So But our unnamed product, this would be the match based on the tree. So any questions on the search? Remember, making trees is basically just for fun, just to see how it's done. The problem with the trees, let me see, is that they changed the site so that you can no longer save the picture easily. Notice when I right click, normally what you would do to save an image, that's not an option here because it's this. And so there's really, I played with it to see if I could easily save the image, but I could not figure out a way to do it unless I create an account with the NCBI, and then that will allow me to save images, but that's not practical to do that. So they made some changes, which in this case were not very good because you can't actually save the image. And and then, yeah, I can't print from here, so I'm not going to do that. But so they've made it more difficult to actually play with the images or save them and use them. So, but you don't have to do that, right? So any questions on what to do? So this is due on Monday. Remember, just submit the one-page document, the answer sheets, not this 12-page monster. And then,
oh, sorry.
yeah.
I was gonna ask, how would you figure out the first thing that gave rise to all those other things in the tree? How would you figure out like the top one?
So what do you mean figure out?
Like how do you like how do you know what gave rise to all of these different things? Like what started it? Like the gray circle all the way left.
We don't know that from here because we just entered 5 different things. So, and we don't have that information. All we can say is that there was a common ancestor in the past that gave rise to these different organisms and their proteins, but there's no way to figure that out from here.
Oh, okay. If you selected all of them, it still wouldn't tell you, I guess.
No, still wouldn't tell you. And that's, it was actually the default was that everything was selected to start with. And that gives you, well, in this case, since we don't have that many options, it's not a much bigger tree, but very often you get at least 100 matches. And so, and here's what we have. But it shows that this is a cluster, right, that's very closely related. And this is a separate group, and then yet a third group. So it does show some divergence, right? But we do know that everything came from a common ancestor. And so we can trace that kind of ancestry. And we know that certain genes and their protein products have been modified by evolution, right, to perform similar and sometimes different functions. But, you know, so it's not unusual to see these clusters, right, are very closely related. But examples, you know, when you look at our arm and hands and compare that to the legs of other animals or the wings of bats, you know, the flippers of whales and dolphins, you see all the exact same number of bones in the same relative position. Some are longer, some are shorter, right? So they've been modified for different functions. In humans, the arm and hands are for grabbing, you know, whales and dolphins for swimming, and birds and bats for flying, but they're the same bones in the same relative position, but they've just been modified for different functions. So, but you're trying to find the original ancestor. We don't know because all those organisms are extinct, right? So that's why it's hard to determine ultimately where everything come from, because those organisms no longer exist. But bioinformatics is a hot field right now. So if anyone's interested in science, in particular, if you like math and computers, then bioinformatics may be something you should look into. So there are many, many programs in bioinformatics and computational biology available. So if you like this kind of stuff, that's worth looking into. All right, so that's all I have for this week. So, like I said, on Monday, lecture exam 4. In lab, it's a review for the lab exam. On Wednesday, we will have a review for the optional lecture final in lecture, and then we'll have the lab exam afterwards. So this is it for new material. Next week is exams and review. And if you don't need or want to take the optional final during finals, then you will be finished by the end of next week. So questions? Okay, that's all I have.